Assalamu alaikum and welcome back to another video. So in this video we are going to talk about microtubules. Previously we discussed about the microfilaments. So let's discuss first and foremost about the microtubule structure and organization. Now talking about the structure of microtubules, it's composed of two subunits. That is the alpha subunit, alpha tubulin, and the beta tubulin. Similar to what we have seen in the previously, we have seen that the microfilaments, they are composed of different subunits, and the component there, the monomeric subunit, which was binding to one after another, that was G-actin, and then it resulted in the formation of a filamentous actin filament, which performs different functions, and that we call as a microfilament. Similar to that, here in case of microtubules, we have different components, we have different subunits, we have proteins, alpha tubulin and then beta tubulin. Now these subunits, these components, these are joined one after another. So see here, so we can have an alpha subunit followed by the beta subunit, then an alpha. So in a consecutive manner, an alpha and beta, they pack together and form microtubule. They form an arrangement. So kind of you can see this structure is formed, this hollow cylindrical structure is formed. Now there can be different organization patterns within a cell. So it's not like you will find just one pattern. Suppose we can say that the microtubule structures which are prominent in the cytoplasm, they occur in the singlet state. Singlet, it means that, so first and foremost, so you can see here it will be an alpha, then a beta, then an alpha and beta. So similarly, all of these together, like 13 filaments, these are formed, they form a hollow structure, and that is a singlet microtubule. Okay, similar to that, we have doublets. So in this case, you will see that the three filaments, these are shared between the two hollow cylindrical structures. So that is a duplet structure, and this type of the organization, it is seen in case of cilia and flagella. The third one, that is a triplet, in which you can see there is sharing between these three, A, B, and C, a hollow cylindrical structure of the microtubules. And this kind of arrangement, it has been seen in case of basal bodies and the centrioles. So let's get back here on understanding the structure of the microtubules. So again, if we have a keen look at the microtubule structure, we can sense that it has a plus end and a negative end. And on the plus end, you will always find a beta tubulin. And on the negative end, you will find the alpha tubulin. Okay. And again, the same thing here, that the beta and alpha, these are bound to GTP bound states and GDP bound states. Now, important thing to remember here is that, so this, if you see here, this one, so the GTP, which is bound to the active site in the alpha tubulin, that is non-exchangeable. So it is as it is. But this GDP, so when it is adding on the plus end, so this GDP, it is exchangeable. It plays a very crucial role in that whole process. So this is as it is, but this one here in the beta tubulin subunit, it changes to the GDP, it gets hydrolyzed and changes to the GDP state. So that was about the structure. Now let's move on to understanding the instability, the dynamic instability, which is seen in case of the microtubules. So now this, first and foremost, what is dynamic instability? Now, if you have a look, so we talked about the polarity of the microtubules. We said that one of the end, that is the plus end, the leading end, at which you will see the rate of polymerization that's very high, as compared to the negative end, where there is more rapidly depolymerization is taking place. The same thing is here. If you start off from here, so these are different beta tubulin structures, okay? Beta tubulin and an alpha tubulin, and you can see here again the beta, it's present, it's bound to the GTP, okay? Now these structures, they assemble together and they lead to formation of this filament like structure, this hollow structure. And again, the second thing is, when this beta tubulin, when it is bound to GTP, you will see this plus end, this leading end, it is, it's in a position that more and more polymerization is occurring at the positive end. So that was the assembly step. The next step is when the disassembly takes place. So you can see this curling up from the plus end. Okay, so that is the catastrophe. And the regaining back the same state as it was earlier, that is the rescue step. So this is the dynamic instability, and this is governed by, it depends upon whether or not there is the presence or absence of the beta tubulin which is bound to GTP. So if it is in a state where it's bound to the GTP, you will see the rescue will take place. But if it's in a state that it's bound to GDP, in a diced GDP, so you'll see the catastrophe taking place. Catastrophe, it means that the plus end, it's now falling apart, so there's the depolymerization at the plus end. Okay, so you can see here, so this is a now here what happens is during the catastrophe, the plus end, it is depolymerizing, and as this disassembly takes place, the GDP beta tubulins, they can be seen. Okay, now let's understand this, where from this whole structure arises. It is basically the gamma tubulin ring complex. It's also called as the gamma Turk. It basically, so if you remember earlier, we have seen in case of microfilaments the three processes. We talked about nucleation as the very crucial step. So this whole process of nucleation, it is the step where actually the assembly of subunits takes place, and then later on different components, different monomeric subunits, they join on the same nucleus. So but important is kind of a basic step, that is the nucleation. So that step, it's regulated, and it starts from a complex that's called the gamma tubulin ring complex, and it starts from the negative end here, and then it will proceed from this gamma complex. You will see here what happens, the alpha subunit and then a beta, alpha beta, alpha beta, so it goes on towards the plus end. On the plus end, you will see the beta end, beta subunit of the tubulin, and towards the negative end, you will see the alpha subunit of the tubulin. So that was about the catastrophe dynamic instability in case of the microtubules. Let's understand different kind of the proteins which are bound or which are associated with the microtubules. Now there are multiple proteins. So since we discussed in the previous slide, we discussed about catastrophe, let's understand what are the different proteins that can be responsible for that. First and foremost, those proteins which destabilize the end of the microtubules. So first and foremost, to the example, it is of kinesin 13. Although rest of the kinesins, they don't have the same role, but kinesin 13, it is an exception. Now here this kinesin 13, what it does is it depolymerizes the plus end. So first thing is that if it is depolymerizing, that means the tubulin, since it's plus end, so on this side mostly the beta tubulins are present, and those beta tubulins, they will be bound in a GDP bound state, not in the GTP state, because it's depolymerizing. So this is the function of the kinesin, which we can see again here. Similarly, similar to these kinesin 13, we have another set of proteins that is called as the stathmins. And they also have the same role of destabilizing the plus ends or the positive ends of the microtubules. So basically, these proteins, they contribute to the process of catastrophe. Similar to this, we have certain other proteins as well, which actually help in the stabilization also. Like we have the EB1 protein. So it helps to recognize the positive ends and binds at those regions. So there are many other proteins which actually help in the stabilization of the plus ends as well. Like we have, these are called as the like we have those tip proteins. So these are different proteins which govern they basically govern the formation, the polymerization and the depolymerization of those microtubule elements.